Okay, let's start. Okay, so हमारा जो टॉपिक चल रहा था, we were talking about the nucleotides. Basically, you talked. Even I had asked the question about the basic structure of the nucleotide. So I don't know why many of you made a mistake. किसी ने फॉस्फेट नीचे बनाया हुआ है, किसी ने पेंटोस शुगर यहाँ पे लिखा हुआ है. So it is a very simple thing. There is a pentose sugar ring, then there is nitrogenous base on one side, there is phosphate on the other side. I don't think it's so complicated, right? And within the nitrogenous base, you have purines and pyrimidines. Today, I will tell you how to remember which are the purines, which are the pyrimidines, because again, in this also, I had asked what are the purines. Some of the students have made a mistake in that question. And a lot of your papers have been checked. I will send your marks in the group. Okay. So this also we had seen that how from the different bases we get different nucleosides, and from
Excuse me,
that we
sir.
will get, yes, right. So we know the different bases will give the different nucleosides and which will give the different nucleotides. This we have already repeated in that lecture. One more question which I had asked was what are the products of pyrimidines, right? So the only difference was some of you wrote amino isobutyrate, they wrote the end products of thymine, but I had actually asked the products of pyrimidines. तो वो गलती करी कुछ स्टूडेंट्स ने, right? And again, this here is where we stopped in the last lecture. We said that there are two types of purine and pyrimidine synthesis: de novo synthesis and salvage pathway. De novo synthesis means we are making something from scratch. Okay, we are taking the rice, we are taking the vegetables, and we are making a dish. Okay, salvage pathway means जो खाना बचा हुआ था, the food which we already have, we are mixing it and we are making another meal out of it. Okay, now for the purine synthesis, we have to see that the different parts of the purine. See, purine is double ring structure, yes? So within the double ring structure, which atoms are coming from where, this we have to know. Okay, so for example, you're seeing this double ring structure, the carbon 4, carbon 5, and the nitrogen number 7, they come from glycine. Okay, so this is the origin, right? So for example, if I see all of you students here, I can ask you कि ये दो तीन students कहाँ से आ रहे हैं, ठीक है? तो somebody is coming from India. In India also somebody is coming from a particular city, then somebody is coming from Pakistan, somebody is coming from Bangladesh, right? So similarly, we have to see which atom is coming from which substance. Okay, so C4, C5, and N7, you have to remember these are coming from glycine. Next, we have the C6. It is coming from carbon dioxide. So these are the origins. Basically, carbon dioxide पे कुछ reaction हुआ तो वहां से ये C6 आ गया और ये कार्बन आ गया. ठीक है, ग्लाइसिन का कुछ ब्रेकडाउन हुआ तो C4, C5 और N7 आ गए. N1 comes from aspartate, and N3 and N9 both come from glutamine. And lastly, the C2 and C8, they both come from N10 formyl tetrahydrofolate. Okay, N10 formyl tetrahydrofolate, or THF. So you have to remember C2 and C8, they both come from this substance. So you have to remember all this because I will be asking you questions. I will choose a particular atom and I will ask you what is the origin of this atom. So you have to remember these. You can simply memorize this picture, this diagram, so it will be easy for you to answer. Okay, now when it comes to pyrimidines, it is a lot simpler. Pyrimidine is having a single ring structure, right? So all of the atoms come from aspartate except one nitrogen, which is coming from glutamine, and one carbon, which is coming from carbon dioxide. राइट, सो यू सी एस्पर्टेट यहाँ भी था, कार्बन डाइऑक्साइड यहाँ भी था, और यहाँ पे भी है, और ग्लूटामीन भी दोनों में है, राइट? ओनली थिंग एक्स्ट्रा इन द प्यूरिन इज ग्लाइसिन एंड एन फॉर्माइल टेट्राहाइड्रोफोलेट. ठीक है, सो दिस यू विल रिमेंबर. प्लीज डोंट ड्रॉ ऑन द स्क्रीन. आई नो आप लोग के सारे बचपन के यहाँ पे वो आ रहे हैं वापस. Okay. Right. Now we are going to talk about the de novo synthesis. Okay. So the two types of synthesis, de novo and salvage. And also, guys, please try to learn the correct spellings. De novo की जगह कुछ भी डी salvo लिखा हुआ है, कुछ भी लिखा हुआ है टेस्ट में. So de novo, novo means new. It's not so difficult. De novo and salvage, right? So when we talk about de novo synthesis, there are 3 points which you have to remember now, and then I'm going to show you how de novo synthesis happens. Okay. So the first point is that the first purine synthesized is inosine monophosphate. So just remember, first purine de novo synthesis में कौन सा synthesize होता है, वो है IMP, inosine monophosphate. और इसका नाइट्रोजनस बेस क्या होता है, वो होता है हाइपोजेंथीन. अब ये क्यों पूछ रहे हैं, जैसे AMP है, तो आई वांट यू टू अनम्यूट एंड टेल मी AMP का नाइट्रोजनस बेस, व्हाट इज द नाइट्रोजनस बेस ऑफ AMP? गाइस सो गए क्या सब?
adenosine monophosphate.
या
No, sir.
बट
Sorry.
AMP का नाइट्रोजनस बेस, व्हाट विल बी द नाइट्रोजनस बेस ऑफ AMP?
Hypoxanthine.
No, hypoxanthine is the nitrogenous base of IMP. I'm talking about AMP. A for
Adenosine.
apple. Adenine will be the nitrogenous base. Okay, so similarly the nitrogenous base of IMP is hypoxanthine. Just remember this is something new. ये आपको याद रखना है, आगे आपको reactions में पता चलेगा कि ये कहाँ पे आते हैं. ठीक है. अच्छा, second question that can be asked is what is the starting substrate in the de novo synthesis of purines? So the starting substrate will be ribose 5 phosphate. ठीक है. एंड वेयर डज डी नोवो सिंथेसिस टेक प्लेस, मतलब ये जो प्यूरिन्स नए तौर पे बन रहे हैं ये कहाँ बनते हैं? एवरीवेयर एक्सेप्ट ब्रेन, बोन मैरो, WBCs एंड RBCs. So this de novo synthesis is taking place everywhere in the body except brain, bone marrow, WBCs and RBCs. What is this message? आर्यन दरवाजा खोल दो. गाइज सुधर जाओ यार, नहीं तो फिर ऑफलाइन आना पड़ेगा. ठीक है. Now you will be able to see all the things that we were talking about, right? Just follow the arrow. Can you all see the arrow? Yes.
Yes, sir.
Okay, so you can see ribose 5 phosphate. So what was mentioned in the previous slide? Ribose 5 phosphate is the starting substrate. तो बात शुरू होती है ribose 5 phosphate से. ठीक है. Then ribose 5 phosphate is converted to phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate. Just these are some new words. Okay, you have to remember this. धीरे धीरे आप 2-3 बार पढ़ोगे तो आपको याद हो जाएगा. तो हमें पता है कि कोई भी रिएक्शन के लिए एंजाइम चाहिए होती है, तो फॉस्फोराइबोसिल पायरोफॉस्फेट बनाने के लिए कौन सी एंजाइम लगेगी? फॉस्फोराइबोसिल पायरोफॉस्फेट सिंथेटेज. ठीक है, सिंपल. उसी उसी सब्सटेंस का नाम लेके उसमें सिंथेटेज ऐड कर दिया. ठीक है. नेक्स्ट ये कन्वर्ट होगी 5 फॉस्फोराइबो 5 फॉस्फोराइबोजिलामाइन, इसमें कन्वर्ट होगी. ठीक है. इसके लिए हमें चाहिए ग्लूटामीन फॉस्फोराइबोसिल पायरोफॉस्फेट अमाइडो ट्रांसफरेज. ठीक है, ये स्टेप उतना इंपॉर्टेंट नहीं है, पहला स्टेप ज्यादा इंपॉर्टेंट था. इसके बाद और बहुत सारे स्टेप्स होते हैं एंड वी फाइनली गेट IMP. ठीक है. इसीलिए हम कह रहे थे कि सबसे पहला न्यूक्लियोटाइड कौन सा मिलेगा, वो मिलेगा IMP. IMP is also a nucleotide. AMP is also nucleotide. GMP is also nucleotide. IMP is also nucleotide. बट इन सारे रिएक्शंस में सबसे पहला न्यूक्लियोटाइड कौन सा मिला, वो है IMP. Now, IMP can convert to AMP also, and it can convert to XMP also, and then XMP will convert to GMP. What are these XMP, GMP, AMP? I will tell you in the next slide. So, 2-3 बातें याद रखनी है. First substrate, please unmute and tell me which is the first substrate in this whole reaction.
Ribose 5 phosphate.
Ribose 5 phosphate. उसमें enzyme एक्ट करेगी फॉस्फोराइबोसिल पायरोफॉस्फेट सिंथेटेज एंड वी विल गेट फॉस्फोराइबोसिल पायरोफॉस्फेट. ठीक है, ये इंपॉर्टेंट स्टेप है. देन वी विल हैव लॉट ऑफ रिएक्शंस एंड वी विल गेट द फर्स्ट न्यूक्लियोटाइड which is
IMP.
IMP is the first nucleotide. Then from IMP, we can convert to AMP or XMP or GMP. Now, one more thing to remember is that if IMP is converting to AMP, the inhibitor itself is AMP. इसका क्या मतलब हुआ कि अगर AMP बहुत ज़्यादा हो जाएगा तो वो खुद ही IMP को इनहिबिट कर देगा. वो बोलेगा बस ठीक है, आई हैव इनफ AMP, no need to convert to AMP. So AMP itself is the inhibitor for IMP and we need GTP for this reaction. Guanosine triphosphate is needed for this reaction. So IMP to AMP, AMP is the inhibitor and there is need for what?
GTP.
GTP. On the other hand, if IMP is converting to GMP, the inhibitor will be GMP. Here GMP, if it is in sufficient quantity, it will say bas ab aur zarurat nahi hai. And the need is for ATP. So ATP will be converted to ADP. Now what are these reactions? Basically these reactions are giving energy. Okay, so when you remove one phosphate from GTP, it will release energy. So that is what is happening over here. Okay, itna samajh mein aa gaya. Now we will go to the salvage pathway. So this was a de novo pathway. Now we will see what is the salvage pathway. So the first question in pyrimidine in the de novo synthesis was where it is occurring. So it was occurring everywhere except a few places like brain, bone marrow, WBC, RBC. This is occurring mainly in extra hepatic tissues. So hepatic tissues matlab hota hai liver, extra hepatic means outside liver. So outside liver salvage pathway for purine synthesis will occur. Okay, and what is the salvage pathway? See, basically adenine, guanine and hypoxanthine. So मान लीजिए बॉडी में कहीं पे कुछ रिएक्शंस हुए और थोड़ा सा एडेनिन बच गया, कहीं से आ गया, ठीक है, तो एडेनिन will convert to AMP, guanine will convert to GMP and hypoxanthine will convert to IMP. ठीक है, मैंने लेक्चर के शुरू में पूछा था AMP का क्या होगा. If AMP is the nucleotide, इसका बेस क्या है? It is adenine, right? So adenine will convert to AMP, guanine will convert to GMP and hypoxanthine will convert to IMP. This is the salvage pathway. हमने क्या कहा था? डी नोवो पाथवे मतलब सारे हमें इतने सारे रिएक्शंस करने पड़े. ठीक है, हमारे पास कुछ भी नहीं था, हमारे पास सिर्फ राइबोस 5 फॉस्फेट था. तो हमने इससे बना के हमने आईएमपी बनाया, एएमपी बनाया, जीएमपी बनाया. दिस वाज डी नोवो. But on the other hand, for salvage pathway, we are using existing adenine, guanine, hypoxanthine, which we already have in the body. We will use certain enzymes. Now, the name of the enzymes are important. Adenine converts to AMP with the help of adenine phosphoribosyl transferase. ठीक है, phosphoribosyl transferase याद कर लो बस, phosphoribosyl transferase. So adenine को convert करेगा adenine phosphoribosyl transferase. बस अब और कुछ नहीं याद करना. Guanine और hypoxanthine are same in the sense that the enzyme required for them is the same. It is a single enzyme which will convert both of these. So the name of the enzyme will be hypoxanthine guanine phosphoribosyl transferase. So this will convert hypoxanthine also, and the same enzyme will convert guanine also. Okay? And for this reaction, we need phosphoribosyl pyrophosphate. We need the PRPP, phosphoribosyl pyrophosphate. It will get converted to inorganic pyrophosphate. Inorganic pyrophosphate. So we are talking about purine synthesis and we are talking about de novo synthesis and salvage pathway. ठीक है, so this was for purine. Purines कौन से होते हैं? Adenine and guanine. तो ये हमने देख लिया purines का. ठीक है, now we are going to talk about pyrimidines. ठीक है, What pathway we have? Okay, before that, within the salvage pathway, see, this is just showing the same thing in a different way. See, follow the arrow. Adenine is converted to AMP with the help of the enzyme adenine phosphoribosyl transferase. इसको short form में हम बोलते हैं APRT. ठीक है, APRT. And as I mentioned, hypoxanthine and guanine, they both use the same enzyme. And the name of that enzyme is hypoxanthine guanine phosphoribosyl transferase. And this is called HGPRT. Okay, the short form of this is HGPRT. Now, salvage pathway, like I mentioned, it is formed from free bases. So body में कहीं पे adenine रखा हुआ था, कहीं पे guanine रखा हुआ था, तो उनको लाके इस enzyme के presence पे हमने easily we are making AMP, IMP and GMP. Now, which enzymes are responsible? So the answer is APRT and HGPRT are the enzymes which are responsible. This is the important point. Salvage pathway accounts for 90% of the nucleotide synthesis. Okay, 90%. So 90% the body is using those things which are already present to create the nucleotides. Okay, and this is used because less energy is consumed in these reactions, because you saw in the de novo synthesis there were so many steps, so many reactions, right? So each step requires energy, but here the reactions are only single step, so the energy required will be less. That's why the body will always prefer कि एनर्जी कम यूज हो. जैसे अभी आप लोग के पास चॉइस था कि आप कहीं पे बैठ सकते थे या आप बेड में लेट सकते हैं, ठीक है? So most of you are already in the bed nicely. So you are saving energy. So that is what we do. We like to save our energy as much as possible, right? Okay. Now I want to show you, basically, I want to create a link in your mind that the de novo synthesis and the salvage pathway, they are kind of connected. Okay. How they are connected? Once we get the inosine monophosphate, which is the IMP, okay, once we get the IMP can itself start the salvage pathway also. Okay. Why? Because when IMP breaks down, we get inosine. Okay. And from inosine, we can get hypoxanthine. And again, we can get IMP, right? So the important thing in this cycle, I'm going to show you, it is covered. There is, just remember, there is a link between de novo synthesis and salvage pathway, right? It's not like in your body only de novo is happening or only salvage is happening. They are both happening. And we told that 90% salvage pathway is happening. The thing that I want to show you over here is that hypoxanthine and guanine, they both convert to a substance known as xanthine. Okay. So hypoxanthine and guanine, they both convert to xanthine, and xanthine converts to uric acid. And this uric acid has to be removed from the body. Okay. Uric acid is excreted from the body with the help of the kidneys. So this is what normally happens. Now, today I'm going to tell you at least 3 diseases. Okay. So every time I talk about diseases and something clinical, मुझे तो बहुत excitement होता है. So I hope you guys also feel excited when we are talking about diseases, right? Because biochemistry is just like, reactions and this and that, but we are trying to learn some diseases also at the same time. Okay. So simple सी बात है, xanthine, I'm sorry, hypoxanthine or guanine, both are making xanthine. This xanthine will make uric acid. Okay. Now, from uric acid, the first disease that I'm going to talk about is uric acid stones. So I don't know if you have heard about this in your relatives or extended family members. Older people, usually they start to get pain in their toe, in the foot, right? And what they are suggested by the doctor is get your uric acid tested. Okay, so the level of uric acid has to be tested in the blood. So that finding may lead to the diagnosis of uric acid stones, and we also call this as Can anybody tell me what we call this, the name of this disease, what we call it?
Gout.
Yes, very good. Right. So, gout is the name of the disease. Now we are going to see what is the biochemistry behind this disease. So I told you that hypoxanthine and guanine, they both convert to xanthine. Xanthine converts to uric acid and uric acid is excreted in the urine. इस मूवी में जो मेन हीरो या विलन, whatever you want to call it, it is the enzyme xanthine oxidase. So hypoxanthine and guanine, they converted to xanthine, everything was good. Xanthine converted to uric acid, everything was fine in the presence of xanthine oxidase. But what happens if this xanthine oxidase becomes overactive, or there is excess xanthine oxidase production in the body? What do you think will happen? Excess, it is working more than normal. Yes? Hello?
Higher level of the uric acid.
Yes, high level of the uric acid. Now, always remember this concept. I was teaching the same thing in the physiology class for first and second semester, that the kidneys have a limited capacity. Okay, so the kidneys can clear your blood from waste materials, but they have a limit. Okay, so even for clearing the uric acid, there is a limit. If there is a lot of uric acid, the kidneys will not be able to clear it. And that will cause accumulation of the uric acid in the joints of the body. Okay. So when it accumulates in the joints, the disease will be called gout. And when it accumulates in the kidney itself, crystals of uric acid in the kidney, then those will form kidney stones. Okay. So problem with one enzyme is causing two different diseases, you can say, or two different symptoms. One of them is gout, which means all the uric acid which was in the body is getting accumulated in the joints. And the second thing is because the function of the kidneys is to clear it as much as possible. So the excess which the kidneys are not able to clear, they are getting accumulated in the kidneys itself. So that will form kidney stones. So these two things are because of xanthine oxidase being overactive or in excess, right? Now, if xanthine oxidase is responsible for these problems, then the solution is also with xanthine oxidase, right? So how can we treat this? What do you think logically क्या करना चाहिए इस प्रॉब्लम को सही करने के लिए? Yes, सो गए सब?
No, no.
Yes.
No, sir.
Inhibit xanthine oxidase.
Yes, we need to inhibit xanthine oxidase. और उसके लिए हमारे पास गाउट के ड्रग्स होते हैं और उसका एग्जाम्पल है एलोप्यूरिनॉल. ठीक है, वी आर नॉट स्टडिंग फार्माकोलॉजी, बट जहाँ पे थोड़ी बहुत एक दो चीजें वी विल ट्राई टू लर्न. कोई नुकसान की बात नहीं है. ठीक है, तो आज से याद कर लें कि एलोप्यूरिनॉल is the drug used for treating gout and it inhibits xanthine oxidase. ठीक है, okay, next disease. पहले दो तीन डिजीजेस की बात करते हैं. द मीटिंग इज गोइंग टू एंड इन 10 minutes. सो आफ्टर 5 minutes आई विल एंड द मीटिंग and you will join a new meeting using the same link. Okay, right, so this is the next disease. And guys, please don't be confused. अभी हम gout की बात कर रहे थे और उसके बाद आ गई ये वाली पिक्चर्स. ठीक है, तो ये gout नहीं है. इतना कॉमन सेंस आपके पास होना चाहिए. आई जस्ट एड पिक्चर्स, सो दैट यू कैन सी द डिजीज, you can be curious about it, and then we will discuss the disease. Okay, so this has happened before in previous lectures. किसी चीज की इमेज किसी डिजीज पे फिट कर दिया आपने अपने दिमाग में. ठीक है, तो जस्ट यूज योर कॉमन सेंस. गाउट यहाँ पे हो गया, जॉइंट्स की बात हो रही है, यहाँ पे कुछ और सीन है. ठीक है, सो कैन एनीबडी टेल मी, हैव यू हर्ड अबाउट दिस डिजीज? व्हाट डू यू थिंक इज हैपनिंग इन दिस डिजीज? क्या हो रहा है? बेसिकली देखने में क्या पता चल रहा है कि व्हाट इज हैपनिंग हियर, व्हाट डू यू सी? हेलो।
Yes, sir.
सर मुंह का कैंसर लग रहा है।
ठीक है, अच्छा गेस है, गुड गेस. Because it's looking like, the lips are damaged, right? The tongue is damaged. Okay, let's talk about the disease. Let's not worry. So this, first, before I tell you the name of the disease and before I tell you the symptom, let's see what is the cause of this disease. Okay. So before I tell you the cause, I want you to go back to these reactions and remember which enzyme was being used. So for converting hypoxanthine and guanine to IMP and GMP, which enzyme were we using? We were using HGPRT, which means hypoxanthine guanine phosphoribosyl transferase. ठीक है, ये यहाँ पे this, you see, hypoxanthine to IMP, this enzyme, and guanine to GMP also, this same enzyme. Now what happens if this enzyme is deficient? Okay, if this enzyme is deficient, then the body starts accumulating lot of hypoxanthine and guanine. So body will start accumulating hypoxanthine and guanine. That is one problem. Second problem is that we are getting 90% of our purines from salvage pathway, right? So this GMP and this IMP which we were getting, it will be insufficient, right? We are not going to get enough GMP and IMP if there is a problem with this enzyme. So if this enzyme is deficient, there will be renal stones, there will be gout, there will be poor muscle control, mental retardation, and the most important feature which you can see, it will be lip or finger biting. So this here, the patients that you see, this is not because of the disease. This is because they are biting their own lips. They are biting their tongue. They are biting their fingers. Okay. So this disease is also called lip or finger biting disease. And the real name is Lesch-Nyhan syndrome. Okay. The name of the disease is Lesch-Nyhan syndrome. So already we have talked about gout. We have talked about uric acid kidney stones. And this third thing is Lesch-Nyhan syndrome. And you have to remember that this is X-linked recessive disease. Okay, X-linked recessive. So X-linked recessive क्या मतलब, ये बहुत कॉमन होगी या ये बहुत रेयर होगी ये डिजीज?
Rare.
Why rare?
क्योंकि सर जेनेटिकली मिली है इस वजह से।
जेनेटिकली तो मिली है, बट रेयर क्यों होगी जेनेटिक के बाद भी?
सर डोमिनेंट नहीं है, रिसेसिव है।
It is recessive, right? So both the parents have to have the gene, and only when both parents are having the gene, it is possible for the child to get this disease. So it is rare, but it is definitely very dangerous disease. Okay. Lesch-Nyhan syndrome. Now, the last disease that I want to talk about is SCID. It is severe combined immunodeficiency. So what are the features? There is high mortality in the first year, which means within the first year, the child might die. Okay. The chances are very high. Absent or low T cells. Just imagine that the immunity is totally crashed. Okay. The T cells and the B cells are either absent or they are very low. They are not functioning properly. And the possible cure will be by bone marrow transplant from a healthy donor. So if you do the bone marrow transplant, I don't know if if you remember this detail, that obviously our cells are formed from the bone marrow, right? Blood cells. So if the bone marrow is transplanted, if the patient gets a healthy bone marrow transplant, then the new bone marrow will start making these cells. So the body will be able to fight infections and things like that. But without the bone marrow transplant, the chances of death are very high. Because just imagine, no B cells, no T cells. The child is not having any immunity. So how is the child going to survive? Let me show you. See, you see this child is inside a big, like a bubble, like a balloon. Okay, so that there is no, like, even common germs, which we can easily, like, every day we are coming in contact with hundreds of germs, dozens of germs, right, every day, but we don't get sick because we are able to fight it. But in this case, it is so severe that you cannot even communicate, right, without a barrier. It has the babies were placed inside protective coverings, such as this. Another example is this. You see, the child is getting the supply for oxygen through the tubes, and the child itself is covered totally from head to toe. The child is even breathing inside that suit. So it is that dangerous, right? Let's continue. So we were talking about severe combined immunodeficiency, which is caused by. So what is the cause of this disease? Adenosine deaminase नाम की एंजाइम है, उसकी डेफिशिएंसी से SCID कॉज होता है, ठीक है। और ये एडिनोसिन डीअमाइनेज आती कहाँ पे है? ये AMP2 आइनोसिन बनाती है, ठीक है। तो ये रिएक्शन हमने अभी तक नहीं देखा of conversion from AMP to inosine, but this also happens in the body. So if AMP to inosine conversion is not happening, then we will see severe combined immunodeficiency, which is the enzyme ADA, which is also known as adenosine deaminase. आगे की रिएक्शन क्या होती है? आगे आइनोसिन से हाइपोजैंथीन बनता है, हाइपोजैंथीन से जैंथीन बनता है। SMA Shakur, please don't draw, my dear. Why you have to draw here? बाद में ड्रॉ कर लेना, अपनी जूम मीटिंग करना, हम लोग सब आ जाएंगे वहां पे, फिर करना ड्रॉ आराम से, ठीक है। So AMP is converting to inosine with the help of the enzyme adenosine deaminase. If this enzyme is deficient, then AMP will not convert to inosine. Inosine will not make hypoxanthine. Hypoxanthine will not make xanthine. तो ये सारी यहाँ से ही अगर प्रॉब्लम हो गई तो आगे भी हर चीज पे इफेक्ट पड़ेगा, ठीक है। So just remember this, and now we can see all these diseases together. Okay, all these diseases we can see together in one frame. So what are these diseases? See, Lesch-Nyhan syndrome over here, Lesch-Nyhan syndrome. It is happening because of the deficiency of HGPRT, right? Hypoxanthine guanine phosphoribosyl transferase. तो ये एंजाइम में प्रॉब्लम होगी तो लेश-नाइहन सिंड्रोम होगा। एडिनोसिन डीअमाइनेज में प्रॉब्लम होगी तो SCID, सीवियर कंबाइंड इम्यूनोडेफिशिएंसी, राइट। सो एडिनोसिन डीअमाइनेज क्या कर रहा था? AMP, यहाँ पे एडिनोसिन लिखा हुआ है but it is AMP. So conversion of AMP to inosine, it will not happen if there is deficiency of adenosine deaminase. And here, if there is excess of xanthine oxidase, so ये ऊपर वाले एग्जाम्पल में डेफिशिएंसी है, xanthine oxidase में excess है. So excess xanthine oxidase will cause gout and kidney stones. So total how many we are seeing? 1, 2, 3, 4 diseases. Okay, we are seeing 4 diseases, all related to purine synthesis. Okay, purine synthesis. All right. Yeah, so this is just once again the whole reaction. One more thing that you can remember is that after uric acid, if uric acid breaks down properly, the substance that will be formed will be allantoin. So allantoin is also normally produced in the body after the breakdown of the uric acid. So that is also one thing you can remember. Now I want you all to read this case. Imagine your doctors, आपके हॉस्पिटल में ये केस आया है, इसके बारे में पढ़िए और बताइए कि इस पेशेंट को क्या प्रॉब्लम है। Which disease? We talked about 4 diseases. Which of the disease fits this case according to you? कहाँ गए सारे डॉक्टर्स? Okay, I am getting some chat. Yes, high uric acid stones, gout. Yes. Okay, good. So this is a case of gout because severe pain in the right big toe, and the patient is saying there is no injury, no trauma. Also, serum uric acid was 9.7, which means it was higher than normal. What is the normal value? You have to find out and you have to write in your notebook the normal value of serum uric acid. That's your homework. Okay, going back to school. All right. So just I showed you this case just so that you understand the importance of this topic, right? Okay. Now, until now, we were talking about purine synthesis, right? We talked about Denovo and we talked about salvage. Now we are going to talk about the pyrimidine synthesis. In pyrimidine synthesis, don't be afraid. This figure is looking very scary, but I'll try to make it simple for you. See, this box down here, this is actually purine metabolism. We have already studied it. आपको देख के लग नहीं रहा होगा कि हमने ये पढ़ा है, but हम पढ़ चुके, ठीक है? These are actually the detailed reactions. So we don't have to study the detailed reactions. We only need to study the main steps of the reactions, right? I want to remove this box from here. Okay, let's see if it goes. No, let's put it here. Okay. So this complete chain of reactions over here, this is actually purine synthesis. Now we have to see pyrimidine synthesis, right? Pyrimidine synthesis. Now in purine synthesis, which was the first substrate? Please say it, unmute yourself and say it. First substrate was? Hello. Are we all still connected with each other? Chat. My mic or your mic? Oh, okay. मैंने ही आप लोगों को चुप करा के फिर मैं भूल गया. ठीक है, you can send a message. Send in the chat. Yes. So ribose 5 phosphate. कितना फनी बात हो गई ना. मैं ही म्यूट करके बोल रहा हूँ कि कोई बात क्यों नहीं कर रहा है. Okay. Yes, good. So ribose 5 phosphate was the first substrate in purine synthesis, right? Then from ribose 5 phosphate, if you remember, we got phosphoribosal pyrophosphate. Now this phosphoribosal pyrophosphate is required in the second step of pyrimidine synthesis. You see this arrow, it is going here for helping this reaction. Okay, so just like ribose 5 phosphate was the first substrate in purine metabolism, in pyrimidine metabolism, the first substrate is carbamoyl phosphate. Okay, so here it was ribose 5 phosphate, here it is carbamoyl phosphate. Carbamoyl phosphate converts to orotic acid converts to UMP. This important reaction I'm going to tell you again, because you see there are a lot of reactions happening here right now. Just remember carbamoyl phosphate याद रखो, ठीक है? This is the first substrate. दूसरी चीज याद रखो that this PRPP, phosphoribosal pyrophosphate, this is also being used. Okay, this is being used. The third thing you have to remember is that what is the end product over here? बीटा एलिनीन, राइट? याद आ रहा है बीटा एलिनीन? मैंने मेंशन किया था प्रोडक्ट ऑफ कैटाबॉलिज्म ऑफ pyrimidine, राइट? उसमें आया था बीटा एलिनीन, CO2 plus NH3 plus beta alanine. So this is actually one of the end products. That's why we are saying that beta alanine is the product of catabolism of pyrimidine. Okay, so this I have highlighted. What you have to remember, you have to remember this PRPP, beta alanine is the end, and you have to remember that carbamoyl phosphate is the first substrate. Okay. Right, see here again I have shown that in pyrimidines we had mentioned carbon dioxide, ammonia and beta alanine. Okay, now In pyrimidine synthesis also, there is one disease which we are going to talk about. Again, disease, I want you all to be excited के disease के बारे में पढ़ने जा रहे हैं. Disease का नाम है orotic aciduria, right? So let's start here. Let's not look at the disease. First, I want you to see कि carbamoyl phosphate first substance था. इसमें जब aspartate add हुआ, see, you can see in the previous slide also. See, aspartate is getting added. So carbamoyl phosphate plus aspartate will make orotic acid. Okay, it will make orotic acid. So before orotic acid, there are a few steps like carbamoyl aspartate banega, फिर dihydroorotate banega. But just remember कि carbamoyl phosphate plus aspartate से बनेगा orotic acid. Orotic acid को orotate भी बोलते हैं. So orotate और orotic acid. Orotate को convert करने के लिए ororidine 5 monophosphate में we need an enzyme called UMP synthase, which means uridine monophosphate synthase. यूरिडीन मोनोफॉस्फेट सिंथेज एंजाइम के एब्सेंस में जो डिजीज होगी उसको बोलते हैं ओरोटिक एसिड्यूरिया. ओके, सो वेरी सिंपली वंस अगेन कार्बोमाइल फॉस्फेट प्लस एस्पर्टेट से बनता है ओरोटेट या ओरोटिक एसिड. ठीक है, ओरोटिक एसिड आगे कन्वर्ट होता है ओरोटिडिन 5 मोनोफॉस्फेट में with the help of the enzyme UMP synthase. UMP synthase मतलब यूरिडिन मोनोफॉस्फेट सिंथेज. यूरिडिन मोनोफॉस्फेट सिंथेज अगर डेफिशिएंट होगी या सही से फंक्शन नहीं करेगी तो ओरोटेट यानी कि ओरोटिक एसिड बढ़ जाएगा. ठीक है, ओरोटिक एसिड जब बॉडी में बढ़ जाएगा तो वो यूरिन में भी निकलने लगेगा. ठीक है, सो दैट इज कॉल्ड ओरोटिक एसिड्यूरिया, व्हिच मींस ओरोटिक एसिड इज कमिंग इन द यूरिन. दैट इज ओरोटिक एसिड्यूरिया, राइट? नाउ इन pyrimidines, that is all we are going to talk about because only this is important, right? In pyrimidines we are only talking about this much in the synthesis. In purines we went into a lot of detail, salvage and Denovo, but in pyrimidine just generally this is all you have to remember, and you have to remember orotic aciduria. After that we are going to talk about DNA and RNA. So ये तो interesting topic है, so I think we can continue. Already, you know, we have missed our lectures. I'm allowing you to unmute yourselves. So let's quickly talk a little bit about DNA and RNA, and then we will continue in the next lecture. Okay. What's the time now? 10:30. Okay. अभी तो 1 घंटा भी नहीं हुआ. Right. So talking about DNA and RNA. See, very simply, RNA is single stranded, DNA is double stranded. RNA is having the helix of sugar phosphate, right? So sugar phosphate helix is present in the RNA also, and double helix is present in the DNA. What is the name of that helix? Sugar phosphate, right? And the nucleobases are present on the RNA, but they are only single. In DNA, they form a pair. Okay, so the nucleobases, what are the nucleobases? Cytosine, guanine, adenine, and uracil. These are the bases in case of RNA. In case of DNA, we have cytosine, guanine, adenine, and thymine. Right? So this is just basic stuff which you already know. Now, the importance of nucleotides. So we were studying all about nucleotides, right? So adenosine, and then adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate. You remember? So in adenosine, we are adding 1 phosphate, it will become adenosine monophosphate. If we are adding 2 phosphates, it will become adenosine diphosphate, and adenosine triphosphate. Similarly, if you add 3 phosphates. The importance is, you remember we say that ATP is the energy currency of the cell. ATP से energy मिलती है. कैसे मिलती है? कि वो एक phosphate को जब अलग करते हैं तो energy release होती है. That's how we get energy from ATP. We break the phosphates one by one. So that is how we get the energy. Okay. So hydrolysis of purine nucleotides
Hello.
gives. Hello. Who's this?
Yes, sir.
हेलो हेलो कौन कर रहा है भाई? इसीलिए म्यूट करा था मैंने. ओके. हेलो हेलो तो जो लेक्चर दे रहा है उसको बोलना चाहिए ना. आप क्यों हेलो हेलो बोल रहे हो? ओके. So you remove one phosphate, there will be energy production. You remove one more phosphate, more energy production, right? So that is the importance of nucleotides. Now, very simply, like you made a mistake, right? Last time, many of you could not write correctly. I asked the purines, but you could not tell the purines correctly. So how to remember that? See, just remember pure as gold rings, okay? So when you get married, when you get engaged, you exchange gold rings, right? So always there are two rings. One the boy will give to the girl, one the girl will give to the boy, right? I hope it is like that only. So two rings, two gold rings, right? Why we are saying pure as gold? Because A stands for adenine and G, gold, stands for guanine. So pure as gold, purines, adenine, guanine, okay? Pure as gold. And two rings, so purines are double ringed structure. You can remember that purines are double ringed structure, right? So for remembering purines, pure as gold. What about pyrimidines? For pyrimidines, you know the pyramids, right? In Egypt, the pyramids, one of the monuments. So you cut the pyramid. So that will give you pyrimidines. Cut stands for cytosine, uracil, and thymine. So these are the pyrimidines. Uracil in case of RNA. So cytosine, uracil, and thymine, they make cut. So cut the pyramid. Pure as gold and cut the pyramid. Okay. So this will help you remember which are the purines and which are the pyrimidines. Right. So next thing, DNA and RNA, very simple. We know that double stranded is DNA, single stranded is RNA. What we need to know is that the DNA is located inside the nucleus. The RNA is mostly outside the nucleus, and RNA is also of different types. We are going to study about that also in the next lecture. The RNA will transfer the genetic information and it will synthesize protein. So main function of RNA is to synthesize protein, and DNA, the main function is to store the genetic information. Okay, so the DNA, the main function will be to carry the genetic information from from one generation to the other. For RNA, the main function will be synthesis of proteins. Okay, and we will see this in detail in the next lecture. We'll go through the whole process. Right now we are just going through the basics of DNA and RNA. Okay, now we say that both DNA and RNA are having pentose sugar, right? Pentose sugar means there are 5 atoms. So you see 1 2 3 4 5, right? In DNA also 1 2 3 4 5. Now I want you to notice that on the left side there is RNA wala pentose, and on the left side RNA wala, on the right side DNA. What is the difference in these two pentose sugars? Can you please tell me? Can you please notice? They're both looking the same.
The OH is replaced by the hydrogen.
Yeah. So in the deoxyribose over here, only this atom is different, right? So hydroxy group is replaced by hydrogen. So basically क्या किया? एक oxygen हटा दिया. ठीक है? That's why deoxy, which means from ribose, we removed one oxygen from this place. That's why it is deoxyribose. Okay. So this will be the only difference, right? Ribose and deoxyribose. अच्छा, यहाँ पे ऑक्सीजन हटा दिया तो हाइड्रोजन क्यों नहीं ड्रॉ किया? Because in this type of structure hydrogen is already assumed. Okay, only if it is non-hydrogen then we have to draw, right? So hydrogen is present over here but it is not drawn. All right, now we are talking about dhire dhire we are trying to make the structure of the DNA and the RNA. Okay, so you see, when you see the helix, the single helix in the RNA or the double helix in the DNA, wo helix kya hai actually? It is these nucleotides are all attaching together, right? So how they are attaching, it is called the sugar phosphate backbone. Sugar phosphate backbone. इसमें sugar क्या है? शुगर है हमारा वो पेंटोज शुगर, राइट? जो हमने बोला राइबोज या डीऑक्सी राइबोज, इसको बोलते हैं पेंटोज शुगर, ठीक है? तो यहाँ पे हम उसको शुगर के नाम से बोल रहे हैं। और जो फॉस्फेट्स होते हैं उसमें, ठीक है, याद होगा आपको, फॉस्फेट एक साइड पे होता है, दूसरे साइड पे नाइट्रोजनस बेस होता है। तो ये जो फॉस्फेट्स होते हैं ना सारे एक साइड पे, ये सब आपस में लिंक करते हैं। सो ऑल द फॉस्फेट्स join together one after the other and they will make the sugar phosphate backbone. Okay, sugar phosphate backbone. And the important thing to remember is that they are all oriented in the same direction, which means फॉस्फेट 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 सब एक साइड पे है और नाइट्रोजनस 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 बेस सारे दूसरे साइड पे है। तो फॉस्फेट नाइट्रोजनस बेस के साथ नहीं ज्वाइन करेगा, फॉस्फेट फॉस्फेट के साथ ज्वाइन करेगा। दैट इज वन थिंग। सेकेंड थिंग इज दैट द फॉस्फेट फ्रॉम थर्ड कार्बन will join the phosphate group will join third carbon of one nucleotide and fifth carbon of the next. Okay, I'll just show you in a minute. I'll explain what that means. See, so we are doing the numbering of these carbons. Okay, so 1 2 3 4 5. Okay, can anybody tell me why this is not 5?
Sir, oxygen.
Yes, it is oxygen. Yes, very good. This is not carbon, this is oxygen. So carbons की numbering इस तरह से होगी 1 2 3 4 5. Okay, so you can see that fifth position पे we have phosphate. ठीक है, ये हमने जब स्ट्रक्चर में बात की थी तो फिफ्थ कार्बन से फॉस्फेट अटैच था, बट वो क्या करेगा, वो पिछले वाले के थर्ड कार्बन से भी अटैच करेगा। ठीक है, so this is how the link is formed. One phosphate will attach to carbon number 3 of the previous one and carbon number 5 of the next one. So that's how this whole chain will be called. And what do we call these bonds? Do you know the name of these bonds?
Phosphodiester, sir.
Yes, yes. So these bonds are called phosphodiester bonds. Actually, why they're called diester? Because there are two ester bonds. Okay, so this from carbon 3 is also ester bond and from carbon 5 is also ester bond. तो एस्टर एस्टर मिला के बन गया डाई एस्टर। ठीक है, और डाई एस्टर में क्या ज्वाइन हो रहा है, फॉस्फेट ज्वाइन हो रहा है। सो दैट्स व्हाई इट इज फॉस्फोडाइएस्टर। ठीक है, phosphodiester bond will be joining them both. Okay, now here I have just shown how they are separated. So nucleotide number 1 2 and 3, here you can see 3 nucleotides in this picture and they are connected. You can see between the first and the second one they are connected by a phosphodiester bond. Okay, now on the other side, इस साइड पे तो फॉस्फेट था, दूसरे साइड पे था नाइट्रोजनस बेस, राइट? Nitrogenous bases are added to carbon number 1. Okay, always remember this. Phosphate is added to carbon number 5 and it will also join with carbon number 3 of the previous one. Nitrogenous bases are added to carbon number 1. Okay, and their order is important to determine the genetic information. So we know the codes, right? GCC, AGC, all the codes will give different, will synthesize different proteins, right? So this sequence is important. We are going to see what are the codons and everything. That also we will see in the future, next lecture. But right now you have to remember that कार्बन नंबर 1 पे अटैच होते हैं, पहली बात। दूसरी चीज, इनकी सीक्वेंस इंपॉर्टेंट होती है। नाउ डीएनए इज मेड ऑफ SMA शकूर, प्लीज म्यूट योरसेल्फ माय डियर, प्लीज डोंट टॉक। पहले तो ऑफलाइन में बोलते थे डोंट टॉक, अब ऑनलाइन में भी बोलना पड़ रहा है। ओके, राइट, सो डीएनए इज मेड अप ऑफ 2 सच स्ट्रैंड्स। तो अभी हम जो पूरी बात कर रहे थे कि एक साइड पे फॉस्फेट है, एक साइड पे नाइट्रोजनस बेस है, तो ये पूरा सिर्फ एक स्ट्रैंड बना। डीएनए में 2 स्ट्रैंड्स होते हैं, ठीक है। तो क्या होगा, प्लीज नोटिस द डायरेक्शन ऑफ दिस पेंटोज। यहाँ पे पेंटोज का शेप ऊपर की तरफ है और यहाँ पे नीचे की तरफ है। ऐसा क्यों? बिकॉज दे विल बी अटैचिंग इन द ऑपोजिट डायरेक्शन। ओके, सो द 2 स्ट्रैंड्स ऑफ न्यूक्लियोटाइड्स, देखो ये एक न्यूक्लियोटाइड है, करेक्ट। ये सेकंड न्यूक्लियोटाइड है, थर्ड न्यूक्लियोटाइड। तो ये न्यूक्लियोटाइड्स जब साथ में अटैच हुए तो इनको बोलते हैं पॉलीन्यूक्लियोटाइड, ठीक है। तो ये पूरा दिस इज 1 पॉलीन्यूक्लियोटाइड, then you're adding 1 more polynucleotide, so 2 polynucleotides, and they will attach in the opposite direction. Okay, and when they attach you will get the DNA. And how they attach, see, some of them will have double bonds, like between adenine and thymine you will have double bond. In between cytosine and guanine you will have triple bond. Which bonds are these? These are hydrogen bonds. So why we are saying, why we are emphasizing the bonds, because you can get a question about the bond which was between the phosphates. Okay, so that was phosphodiester bond. And which is the bond between the nitrogenous bases? It is the hydrogen bond. So you have to remember that these 2 bonds are different. Okay, again the meeting is going to end in 10 minutes. Oh my god. All right, so this, like I told you, the sister strands of the DNA. So these 2 strands, these 2 polynucleotides, they run anti-parallel, which means in opposite directions. They are joined by the bases. A is always paired with T, G is always paired with C. Okay, ATGC. Just remember, A will pair with T, G will pair with C. And purine will always be paired with pyrimidine. Okay, so A, G क्या थे? प्यूरीन्स थे, और C और T क्या थे? pyrimidines थे। तो प्यूरीन प्यूरीन कभी भी आपस में नहीं पेयर होगा। प्यूरीन will always pair with pyrimidine. The sister strands are complementary but not identical. What does this mean? So if we look at one strand, we will be able to tell the other strand. Why? Because A जहां पे होगा उसकी जगह T होगा, वहां पे G की जगह पे C होगा, right? So we can tell what is the opposite strand, but it will not be the same. It will not be identical, right? So they are called complementary. And these bases are joined by hydrogen bonds. We've already mentioned they are joined by hydrogen bonds. Okay, we are almost going to finish the lecture now. So yeah, just remember each purine is paired with pyrimidines. Now here, once again, the same image we have zoomed in, and when you get the PDF, you can see, you can understand. Right. Now in this picture, I want you to notice that, just look here on the left side, you see the 5 carbon is up, the 3 carbon is down. If you look at this chain, and here the 3 carbon is up, the 5 carbon is down. So we call it the direction. We call it the direction. So this will be called the 5 to 3 direction, and this will be called the 3 to 5 direction. Okay, we will talk about it in detail, but right now just try to understand what we are going to say. So it is the 5 end and the 3 end. Okay, so 5 end, 3 end, 3 end, 5 end. मतलब we are trying to recognize कि कौन सी साइड कहाँ पे है, ठीक है ना? We are trying to understand that. That's why we are given the names 5 end and 3 end. Same thing you can see in this picture also. This is the 5 end, this is the 3 end, and on the opposite side, this is the 3 end, this is the 5 end. Okay. So why do we say this? Obviously because of the location of the carbon atom, right? So if you place it in this way, you can see here will be the 5 and 3 end. So if you make a chain out of it, so it is going to end at either the chain will end at 5 or it will end at 3, right? So it has to be one of the two. and this is used for the direction of the DNA strand. ठीक है, so we are able to say कि DNA किस direction में है. ठीक है, अगर 5 to 3 बोल दिया तो we know कि 5 से शुरू हुई बात और 3 पे खत्म हुई. Why we are saying this? Because in the body, in vivo means in the body, the DNA is always synthesized from 5 to 3 direction. Okay, 5 to 3, never 3 to 5, 5 to 3. तो जो DNA synthesis शुरू होगा 5 end से 3 end तक जाएगा. ये बात इंपॉर्टेंट होगी जब हम सिंथेसिस डिटेल में पढ़ेंगे. वी आर गोइंग टू स्टडी सिंथेसिस इन डिटेल. राइट नाउ द रूल इज इंपॉर्टेंट दैट डीएनए सिंथेसिस in the body always happens in 5 to 3 direction. Okay, now the double helix. Who discovered the double helix? Do you know the name? Do you remember the name? School में पढ़ा होगा.
Watson and Crick.
Yes, Watson and Crick ने डिस्कवर किया था. और ये है वॉटसन और क्रिक, इनसे मिलिए. Watson and Crick, these are my students, these are Watson and Crick. Okay. And that's all for today. Now next lecture we are going to study in detail about the DNA synthesis and RNA and everything. Okay, that will be the next lecture.